Thank you, Dr. Aird, for that great talk to get us going. I'm Jerry Luddy from the Wilmer Eye Institute at Johns Hopkins, and it's my pleasure to introduce the second speaker, Shayn Peirce Cottler, who will talk about the secret to growing microvascular network. It's all about forming the right connections.
While we're getting set up here, I just want to thank the organizers for putting together what looks like a really interesting and stimulating conference. And I'd also like to thank the organizers of the session for inviting me to participate as a speaker. So my name is Shayn Peirce Cottler. I'm in the Biomedical Engineering Department at the University of Virginia. I'm also in the Cardiovascular Research Center. And as shown in an earlier slide, I'm a member of the Council of the Microcirculatory Society, and I'm currently serving as art treasurer. In 360. Yeah. Thank you. Okay. So I want to start out with acknowledgments. I have a number of people to acknowledge for all the work I'm going to talk about, and I'm also going to keep this at a very high-level overview. Members of my lab, one of whom is here today, Molly, my collaborator in ophthalmology at UVA, Dr. Paul Yates, my close collaborator at Tufts University, Ira Herman, at UNC Chapel Hill, Vicki Bouch, at Johns Hopkins, Phelan McGowan, Virginia Tech, John Chappell, at Yale, Jay Humphrey, and at University of Texas, Dallas, Heather Haenga. And fortunately, as I said, a lot of these people are here today. So if I say anything that sounds interesting, you're welcome to follow up with those who are attending. So we're interested in studying the microcirculation in my lab, and we're particularly interested in growth and remodeling. We approach this specifically by using a combination of experimental and computational models to understand how microvascular growth and remodeling plays out and how it might be altered in disease. And so we use these tools to try to figure out what the levers and switches and knobs are that we might be able to turn using drugs or cell-based therapies. So just to orient you, and is it possible to get the lights down a little bit? I have a lot of pictures and movies that will show up better if there's a dark room. Is anybody standing near a light switch? Am I? Scared of pushing buttons. This looks really complicated. Okay, well, we'll go on with it. So just to orient you to the types of things we look at in the lab on a day-to-day basis, we tend to be very, oh, that's perfect, thank you very much, very tissue agnostic. We look at a lot of different tissues, and we look at the whole microvascular network structure. So this is a micrograph from the rat mesentery. You can see the scale bar down at the bottom. And what I want to impress upon you is the fact that it's a very complex structure. Of course, we have the arterioles and we have the venules shown over here at the top left. Let's see if my mouse will work. There are a few branches off the mesenteric artery and the mesenteric vein, and they come into this tissue, and of course the arterioles branch off further and form the capillaries, and these are the structures shown here in green, these sort of green tendrils, which then ultimately come and link back up to the venules. You can see here. So this is the rat mesentery. The pattern looks quite different in the mouse retina. Here we're looking at a whole retina, and you can see the central arteries and the central veins come in at the optic disc, and they have this sort of radial pattern. They kind of alternate artery, vein, like the spokes on a bike wheel, for example. And in between these, we have the capillary networks connecting. In the mouse ear, we also see somewhat of a different pattern. We see the arterioles and the venules coming in, and they branch together, but they course together for quite some time, down to many levels of branching before they radiate out into the capillary beds that ultimately connect them. And then last but not least, we have a skeletal muscle shown here. This is the mouse spina trapezius. And we see, again, a lot of branching. We have arteries and veins being paired as they enter the tissue. They come down to the level of terminal arteriole and the terminal venule, and then they branch into capillaries, which are invisible here at this level of magnification. So when we look at these different patterns and different types of tissues, the obvious question is, how did they form and how do they adapt during different diseases? So microvascular growth and remodeling is a complex collection of different morphogenic behaviors. The two that I'm going to focus on today are angiogenesis and arteriogenesis. So this is a lovely movie from my collaborator at North Carolina, Vicki Balch, of an embryoid body that's spontaneously undergoing angiogenesis. And you can see very nicely endothelial cells dynamically protruding into their space and connecting up with one another to form a nascent vascular network. So angiogenesis, again, it's new capillaries sprouting from parent vessels. Arteriogenesis, on the other hand, another aspect of microvascular growth and remodeling, is when arterioles and arteries enlarge. And this is work from Phelan McGowan showing that in a spina trapezius muscle, if you induce a ligation or an ischemic event, over a short period of time, you can actually get enlargement, and that's depicted here with these red arrows of some of the feeding arterioles that perfuse this tissue. So this is at an early time point immediately after ligation, and this is a few days later, and you can see the diameter of these vessels pointed out with the red arrows almost doubles. So one thing I want to impress upon you is that these remodeling events happen on a very short time scale. In the case of angiogenesis in this embryoid body model, it can be on the order of hours. And in the case of this muscle, on the order of days. Now, granted, this is in a mouse, and it's questionable as to how this translates to humans, but even if we're at the same order of magnitude, it still suggests that these processes are very dynamic and can happen quite quickly. And of course, angiogenesis, from the disease standpoint, is implicated in diabetic retinopathy, cancer and tumorigenesis, and endometriosis. And arteriogenesis is usually affiliated with the ischemic disorders such as stroke, cardiovascular disease, and peripheral vascular disease. So just to impress upon you the cell biology, we typically think of angiogenesis being a result of endothelial proliferation and migration, and arteriogenesis being the hyperplasia and hypertrophy of smooth muscle cells that line the arterioles. Okay, so I really have just 3 points that I'd like to make. The first key point is that the structure of the network matters when it comes to forming the right connections or growth and remodeling. The second point is that composition matters. And the third point is really that the integration of these two across spatial and temporal scales matters. So point number 1, how and where vessels connect to each other really affects angiogenesis. And I mean no disrespect to our earlier speaker, but it's not all just capillary soup. And that is a notion that I find comes out in conversations with my clinical collaborators, particularly in cardiology, ironically. So this is some work that we did a while ago in collaboration with Dr. Murphy down at Tulane. And basically we showed that there is polarization or heterogeneity in expression of perivascular NG2, which is neuroglial antigen 2, which is a proteoglycan expressed by smooth muscle cells and pericytes. So this is the rat mesentery again. And what we see if we zoom in is that there's expression of NG2 on the arterioles, but it's completely absent in veins, at least in the quiescent network. What's really fascinating, though, is that when you induce a stimulus, this polarization of expression of NG2 changes very rapidly. So just after 3 days after a wounding event in the mesentery, we see upregulation of NG2 along venules, where previously venules that were not expressing NG2. And along with this upregulation, we also see an increase of incidences of capillary sprouting that you can see here. And we'll zoom in, you can see again, venules now expressing NG2 in the smooth muscle cell layer and along pericytes, and also this is commensurate with the capillary sprouting that's coming off of these venules. We think this might be important because about 90% of the time we see the capillary sprouting occurs from the venule tree as opposed to the arterial tree. So again, in terms of arteriovenous polarity, it's important to appreciate because it has implications on where angiogenesis can happen in a network. The structure of the network also matters when it comes to arteriogenesis. And so this is work done by Phelan a few years ago when he was in my lab, and he was looking at the spina trapezius muscle of 2 different strains of mice. On the left, you'll recognize a C57 black 6 mouse, and on the right is a BALB-C mouse. Two different inbred strains, same muscle, but very different pattern of network, especially when you look at the arteries and arterioles. So you can see on the left, we get a lot of interconnecting loops in the arterial tree. And on the right, in contrast, it's much more of a dendritic structure in the BALB-C. And Balaam showed that this is important because if we ligate one of the feeders that provides oxygen and nutrients to this muscle, the remodeling that happens is very different depending on the structure. So in the C57 black 6, this is where we get what we call the arteriogenesis in those loops, those interconnected loops. And in the BALB, the arteriogenesis happens at interconnections between the arterial trees, but these are occurring at the capillary level. And you can see that here, where capillary-sized vessels are recruiting smooth muscle cells that are shown in red in response to the ligation and alteration of blood flow. So here, the key point is that the location of pre-existing connections between arterioles affects where arteriogenesis can happen, either in the big vessels, if that's where the connections are, or in the capillary-sized vessels. So along with this, if you've been paying attention, you've probably noticed these blue arrows, which represent the paired venule in this particular network. And we showed recently that it matters whether or not a venule is next to a remodeling arteriole, because it affects the recruitment of monocytes and the extent to which they can come into the tissue to affect arterialization. And so this is a time course. Sorry, it was a time course. There we go. It's a time course, again in the spina trapezius muscle, where we've ligated that upstream feeder. And you can see after 6 hours, those green cells on the left are infiltrating into the lumen of the venule and adhering, we think. And then after 24 hours, they're starting to extravasate into the tissue. And by 72 hours, these cells are really collecting, they're sort of making their march over to the arteriole that's undergoing remodeling and collecting there. And so, again, the key point is whether or not that arteriole that needs to remodel is close to a venule may affect the extent to which arteriogenesis happens in that vessel and how quickly it might happen. Okay, so key point number 2, composition matters. So the heterogeneity of cell types and their interactions with one another affects growth and remodeling. And it's not just in endothelial tubes. There are other cells that interact with each other, such as pericytes and microglia in the mouse retina, for example, that you can see here depicted on the left, all in close proximity and likely transducing signals to one another to affect how angiogenesis will occur in this tissue. So that's a healthy retina. And on the right here, we're showing endothelial cells labeled with a dye in a pathological retina. This is a Kimba mouse, which is hyperglycemic and also has pathologically elevated levels of VEGF. And as a result, we see what is commensurate with diabetic retinopathy phenotypes in humans, which is vascular dropout, as indicated by these red perimeters. So we found that we can actually affect the morphology of this network or the structure of this network by injecting cells. And you can see the result of an intravitreal injection into the contralateral eye of the same mouse that has disease when we've provided human adipose-derived stem cells. And what's interesting is that we don't think these cells are becoming endothelial cells. We do think instead they're becoming perivascular support cells or pericyte-like cells, which are affecting the endothelial structure of that capillary bed and the pathological retina. I should also mention there's some very nice work recently published by my collaborator Ira Herman that showed that in fact the cytoskeleton and the mechanotransduction capabilities of the pericytes can very much affect the phenotype of the endothelial cells. So there's a lot of molecular biology underpinning the fact that pericytes and other cell types can very strongly affect endothelial cell morphogenesis. And also, as was mentioned too in the previous, as was mentioned earlier, there's a suggestion that these pericytes can also be a source of stem cells, which is also very provocative. Oh, 1 minute. Okay, sorry. So this is a good place to stop and summarize. When you put it all together, the structure and the composition and how it affects function and remodeling, I think it's important that we broaden from our experimental tools and also include computational tools. And that's what we're beginning to do with the collaborators shown here on the left. So the idea of simulating these cells in space and time, where we can simulate individual endothelial cells and pericytes and collections of those cells, and also the growth factors and the receptors that bind growth factors, in order to increase our repertoire of tools that we have for trying to synthesize and integrate across scales. So the lessons learned, structure matters, identity matters, and certainly dynamics. And moving forward, I think the tools that are going to help facilitate our learning in this area include computational modeling, but also non-invasive imaging, high resolution. Thank you.
Jerry Meininger, I have one question for you. There's a lot of emphasis on angiogenesis and arteriogenesis. How important a role is vascular regression in pruning
Critical.
a network?
Absolutely critical, yeah.
Because it's clearly this happens in many disease states. And
Yeah.
there must be a whole host of mechanisms that are just as important for that.
Absolutely. I completely agree with you. And I intentionally focus on angiogenesis and arteriogenesis, but not at the exclusion of vascular regression, intussusception, and other modes of vascular growth and remodeling. Thank you.
John Lutter at University of Maryland. I was interested in the parasites on the one of the later slides you showed, with the parasites being highly populated, I think around the endothelial capillaries, maybe?
Correct.
How important is their contractile function in these developments?
Extremely important, and I would reference you to my collaborator sitting there in the white shirt, Ira Herman, who studied that incredibly well and has a great publication just came out in American Journal of Cell Physiology last month.
Manfred Böhm, over here. Manfred Böhm, Heart, Lung and Blood Institute. Excellent talk. I was wondering, when you show this embryonic body and the capillary network formation in there, this capillary network is forming without any blood, right? It's just forming in. And I was wondering if you can comment on a very different function of endothelium, not only to be the wall of the blood vessel, the inner lining, but also to instruct other tissue or the other cells to become what they are. Like, I think it's important for hepatocyte maturation, for liver cell, for lung cell maturation, and hematopoietic maturation is completely independent of its function to keep the blood in the vessel.
Absolutely. I think that's a wonderful point as well. It's important not to consider the vessels in isolation, but also include the tissue in which they're interacting. So the myocytes and muscle and hepatocytes and so on and so forth. Yeah, there's a lot of crosstalk going on that's affecting the phenotype of the vessels, but also the vessels are affecting the phenotype of those stromal cells in the tissue. Absolutely.
Very nice talk. When you address the connection or composition using the cell perfusion, maybe stem cells, you show the parasites. I find this very interesting. Would you imagine that the stem cell or progenitor cell perfusion would firstly contribute to the endothelial cell or arteriogenesis instead of forming the surrounding parasites, which may mostly contribute to the vascular remodeling, which may be a pathological consequence? Would that be a very serious issue to be avoided?
Yeah, I think it's absolutely important to consider the crosstalk of pericytes with endothelial cells. There's a long line of evidence, particularly in diabetic retinopathy, that implicates the pericyte endothelial connection, and perhaps, not just whether or not the pericytes are present, but how functional those pericytes are in disease and their ability to modulate angiogenesis.
Thanks very much. For the sake of time, I think we need to move on.